it's now Lindsay, who is going to introduce her work on machine learning on adaptive immune receptor repertoire for the discovery of disease biomarkers and predictors in cancer.
Yes, thank you for that. So yeah, I am going to, first I want to thank the panel for inviting me to present. It's great to be able to present in this forum. So I'm just going to quickly give background information. Maybe the only people in this session are people who are as obsessed with repertoires as those of us in the air community are. But in case there are some people from outside the community or even non-immunologists here in the session, I want to just briefly give some introduction to that. And then I'll introduce our research question, describe our computational approach, and show the results that we have within one specific application area, and that is in exploring the potential utility of T-cell receptor repertoire-based biomarkers for cancer early detection and diagnosis. And then hopefully I'll have time to end in the spirit of this session by talking about public sharing of adaptive immune receptor repertoire data. So the most common type of T cell expresses a T cell receptor that is a heterodimer of an alpha chain and a beta chain. The genes encoding both proteins are somatically generated in developing T lymphocytes by a DNA recombination process called Bdj recombination. In the germline, the antigen receptor loci contain non-contiguous gene segments. So here I'm showing the beta chain locus on the left and the alpha chain locus on the right. And we see two gene segment types at the alpha locus, V in red and J in yellow, and then three gene segment types in the beta locus, V and J, and then the third type, D in green. And at both loci during Bdj recombination, one gene segment of each type is selected essentially at random and joined with the others to form the gene. And in addition to this sequence diversity that's created through this process of mixing and matching different gene segments, there's additional diversity created at the junctions by exonucleolytic activity and the addition of non-templated nucleotides. So each developing T lymphocyte has essentially a unique gene sequence at these two loci. And based on the number of gene segments of each type in humans and these patterns of junctional diversity, it's estimated that the total potential alpha beta T cell receptor diversity is around 10 to the 18, although I think it was either Incarnita or Victor presented that at any point in time, the number is maybe closer to 10 to the 8. So this somatically generated region of the gene encodes the part of the beta chain or alpha chain that forms the antigen binding site of the T cell receptor here, and that is referred to as complementarity determining region 3 or CDR3. And in principle, each T cell receptor has specificity for a different antigen than other T cell receptors, and this tremendous T cell diversity, repertoire diversity, means that there should be a T cell receptor with specificity for essentially any antigen that might be encountered. After undergoing Bdj recombination for T cells, this is followed by selection in the thymus to ensure proper TCR function. And then the lymphocytes exit the thymus and crawl around the body, sampling the antigens they encounter. And then upon encounter with their cognate antigen, they will proliferate and create a clone of cells bearing identical T cell receptors. And this is referred to as clonal expansion. So the full set of T cell receptors in an individual at a specific time is referred to as the T cell receptor repertoire. And due to clonal expansion, it bears an imprint of both past and current immune responses. And deep sequencing of the T cell receptor encoding genes of these CDR3 sequences allows a detailed characterization of the repertoire in a particular sample by allowing us to enumerate the T cell clones that are present in the sample, to quantify their relative abundances, to estimate the diversity and clonality of the repertoire, where clonality is a measure of the number of expanded clones and their degree of expansion, and to track clones over time and between tissues. And through that, we are able to make inferences about population level processes such as migration and clonal expansion. So I'm going to focus on one specific area of research in my group, which is to identify patterns in immune repertoire deep sequencing data that are associated with sample or host level phenotypes. So I'm going to talk about work in cancer using T-cell receptor repertoires to identify tumor-associated T-cell receptors and to explore the feasibility of their use for cancer early detection and diagnosis. There are other interesting and useful applications of identifying tumor-associated TCRs in the clinic, but I won't have time to talk about those today. So we hypothesized that detecting anti-tumor T-cell responses by leveraging the specificity of T-cell receptors might facilitate cancer detection and diagnosis. And so we set out to determine the feasibility of this hypothesis by asking a series of questions. So first, can TCR repertoires associated with cancer tissue be distinguished from TCR repertoires associated with healthy tissue of the same type? So can TCR repertoires in ovarian cancer tissue, for example, be distinguished from TCR repertoires derived from healthy ovaries? Two, do different cancer types have different TCR repertoire patterns? So for example, can breast cancer associated TCR repertoires be distinguished from ovarian cancer repertoires? And three, can cancer associated repertoire patterns be detected in the blood? And four, can TCR repertoires associated with cancer tissue be distinguished from TCR repertoires associated with benign masses of the same tissue type? So if a patient presents with an ovarian mass, can TCR repertoire patterns help inform whether the mass is benign or malignant? So we set out to answer the first question using publicly available data described in this publication from Sherwood et al. And the TCR sequencing was conducted by Adaptive Biotechnologies, and the data are available from their public data portal, Immune Access. These data are from 15 patients diagnosed with colorectal cancer, although sequence data are only available for 14 of the patients. The basic study design is that from each patient, there are two samples, tumor tissue and adjacent healthy tissue, and the adjacent healthy tissue serves as the control. And the goal for each sample is to identify CDR3 sequences that allow accurate prediction of whether the sample is a tumor-derived sample or a healthy tissue derived sample. And this is done by identifying individual TCR sequences within the repertoire that serve as a biomarker indicating that it derived from a tumor-infiltrating lymphocyte population. So to accomplish this, we're taking the following approach. So first, we're extracting all amino acid sequences of length 4 from all the CDR3 sequences in a patient sample, and we refer to these as snippets or k-mers. We then convert each snippet into a physicochemical representation using Ashley factors. And finally, we apply leave one out cross-validation to develop a model that will allow us to predict the sample level labels. So whether it's a tumor sample or a healthy tissue sample, to make this prediction from the set of snippets associated with that sample. So we score every snippet within the sample and then aggregate the scores to assign the sample level label. So just more concretely, so we extract all the snippets of length 4 from all the CDR3 sequences in each sample. And then we convert the amino acid sequence of the snippets into a physicochemical representation. And this is an important component of our method because we believe this enables us to capture the physicochemical similarities between distinct amino acid sequences that may be allowing different amino acid sequences to interact with the same antigen. So for example, arginine and lysine both have positively charged side chains. So we use Ashley factors for the representation, and there are 5 Ashley factors that roughly correspond to hydrophobicity, secondary structure association, size, codon usage, and electric charge. So for example, take the snippet arginine, tyrosine, arginine, aspartic acid. We create a numerical vector representation of the snippet by taking the 5 Ashley factor values for each of the snippet's amino acids. And this results in a vector of length 20 that then is the representation of that snippet. So now for each sample, we have basically a bag of snippets, but in Ashley factor space rather than amino acid sequence space. And we then want to apply machine learning to learn labels at the level of the sample rather than at the level of the snippet. And this approach of learning labels for sets of objects rather than individual objects falls under a machine learning framework called multiple instance learning. And so to assign these bag level labels or sample level labels, we have to aggregate the scores in some way after scoring every snippet. And in the results I'm presenting today, we simply took the maximum scoring snippet and assigned the sample level label on that basis. So we applied our method to determine whether we could correctly classify each sample from the colorectal cancer patients as being either tumor or healthy. So here we see patient on the x-axis, and on the y-axis we see the probability calculated by our model of a sample being a tumor sample. So a probability of 0.5 is the threshold, with samples scoring higher assigned a tumor label and samples scoring lower assigned a healthy label. The green points correspond to true healthy labels and the red points correspond to true tumor samples. And we get an overall classification accuracy of 93% by leave one out cross-validation, where all the tumor samples are correctly classified, although 2 are very close to this threshold. 12 of the 14 healthy samples are correctly classified. And it's important to note that the tumor and healthy samples from a single patient are scored independently, meaning the model has no knowledge that the 2 are from the same patient and that one must be healthy and the other must be derived from a tumor. So it's good that even though these 2 healthy samples are misclassified, that in both cases the patient matched tumor sample does receive a higher score from our model. We can look at the model weights to determine what snippet features result in assignment of a tumor label, and we see an increased probability of a colorectal cancer classification for samples containing TCRs with 4 small positively charged residues that appear frequently in alpha helical segments, with 2 hydrophilic residues flanked by 2 hydrophobic residues. We examined the CDR3 sequences that contain the high scoring snippets. The CDR3 sequences are shown here with the high scoring snippets aligned and highlighted in red, and the rank is shown here where 1 corresponds to the most abundant clone in a sample. And interestingly, many of the CDR3s are not among the most abundant clones in the sample. So we next applied the same approach to tumor and adjacent healthy tissue from patients with early stage breast cancer, and these data are also available from Adaptive Biotechnologies portal and are described in this publication. And there are 16 patients and their clinical information is shown here. And as with the colorectal cancer data, we found that all tumor samples were correctly classified. There are 2 misclassified healthy samples and 2 additional healthy samples that score close to the threshold, but are technically correctly classified. For all 4 of these, the corresponding patient matched tumor sample received a higher score, and the overall classification accuracy is 94%. Again, we can analyze the model weights to determine what CDR3 snippet features are associated with the sample receiving a breast cancer label. And we find a different pattern than that observed for colorectal cancer, where in this slide, the colorectal cancer motif is shown in the top panel and the breast cancer motif is shown in the bottom panel. And note, for example, the differences for Ashley factors 2, 3, and 5. Again, we analyze the CDR3 sequences containing the high scoring snippets, and in contrast to what was observed for colorectal cancer, in this case, we find that most of the CDR3 sequences are associated with the most abundant clones in the sample. So if we look back to our series of feasibility questions, we can see that we can answer yes, or at least a tentative yes to the first 2 questions. So in this pilot study, using publicly available data, we have demonstrated that tumor-associated TCR repertoires can be distinguished from adjacent healthy tissue-associated repertoires on the basis of TCR sequence patterns. And we have demonstrated that at least for breast and colorectal cancer, there are two distinct cancer-specific motifs. So what about our third question? Can cancer-associated repertoires be detected in blood? So TCR deep sequencing data from peripheral blood of the 16 breast cancer patients is also available in Immune Access. So we determined whether the motif-bearing CDR3s from the patient's tumor repertoires were present in the patient's peripheral blood repertoires. And we found that they are for 15 of the 16 patients. And overall, we found 19 of the 22 motif-bearing CDR3s in the corresponding blood repertoire. So now we can also answer at least a tentative yes to the third question. So what are the caveats of the study? So the sample sizes are relatively small, which is appropriate since this was just a proof of principle study. But there are two big caveats that still must be addressed for sufficient proof of principle to justify a large-scale study. And the first is we need true control samples instead of adjacent healthy tissue samples. And we need an independent test data set on which to evaluate the model's true performance using data that was not used at all for model selection and parameter fitting. So we use these results to justify funding for a study in which we sequenced repertoires from patient samples collected at UT Southwestern Medical Center, which is my institution. We conducted the pilot study in ovarian cancer, a cancer type for which early detection methods are urgently needed. We collected tumor samples from patients with high-grade serous ovarian carcinoma, and we collected healthy ovarian tissue from cancer-free women. So in this study, we have true control data. Additionally, we sequenced samples from 20 patients in each group, so 40 total, and set aside half to use as test data. So diagnoses for the 20 patients used for the training data are shown here. And the results, we see model performance by leave one out cross-validation on the same order as the other two studies, so 95% with one misclassified tumor sample and one healthy sample that, although it's technically correctly classified, its score is very close to the threshold, so indicating that the model was, uncertain in its classification. A look at the model weights is shown here, and this motif is different from the ones shown for breast and colorectal cancer, so it's a bit too crowded to show the full motif from all three models on a single slide. But here you can see for the first Atchley factor, I'm sorry, that slide didn't advance. The first Atchley factor hydrophobicity, you can see the breast cancer motif here, then the colorectal cancer motif in the center, and then the ovarian cancer motif there on the right. So here we see the diagnoses for the 20 patients used as test data. So these repertoires were not used at all for model selection and parameter fitting. And model accuracy here is 80%. With one misclassified healthy sample and 3 misclassified tumor samples. I think this drop in performance is expected when moving to test data, particularly given the relatively small size of our training set. So having provided now much stronger support for the first two questions, that tumor versus healthy tissue associated repertoires have distinguishing patterns that appear to be specific to the type of cancer, we turned our focus to the last question of whether repertoires associated with benign versus malignant masses can be distinguished via their TCR repertoires. So the same study design, although rather than collecting healthy ovarian tissue from cancer-free women, in this case, we collected samples from women presenting with benign ovarian masses. Their diagnoses are shown here. We have, again, 40 patients where we used 20 for training data and 20 for test data. The classification accuracy in this case was 90%. We used here the benign mass as a positive label. Here I'm just quickly showing diagnoses for the test data. And the classification accuracy when we move over to the test data drops down to 75%, but still some signal that is generalizing to unseen data. So now we can answer at least a tentative yes to this last question, that TCR repertoires associated with cancer can also be distinguished from repertoires associated with benign masses. So I've shown the data from this table that is inside this red box. We're quite excited by the results. So permutation analyses consistently achieve much lower classification accuracies with an average of around 49%. And even though the model performance decreases as we move from the cross-validation results over to the test results, we're excited that given our small training set sizes, we find signal that generalizes to unseen data. And so we are now in the early stages of a large-scale study in which we're recruiting 600 patients with patient-matched tissue and blood, and a focus on patients with early stage disease. So maybe in the 2022 meeting, I can present those results. Given the airr community focus on public sharing of data, I wanted to highlight that all of the data used in this study is publicly available. So the first two data sets from Adaptive Biotechnologies, the ovarian cancer data for which we did the sequencing, we've made that publicly available. And I would like to call out the airr data commons, which I think there's a whole presentation dedicated to airr data commons later. But if you have interest in sharing any airr-c data that you are generating, or if you would like to use airr-c data in the ways that I have, generated a hypothesis and assessed feasibility using public data before moving to the stage of spending a lot of money on sequencing, you can access airr data commons data for those purposes. And we're happy to help. You can email any of these email addresses to get help with sharing or accessing airr data commons data. And with that, I'll just close with acknowledging my funders and collaborators. And most of this work was done by Jared here and Scott. Thank you, and I hope I didn't go over.
Thank you, Lindsay. So I just realized that there's a 1 minute delay between the Zoom and the live. So I don't know if anyone has a question here. Meanwhile, people are listening to the end of the presentation. I have a question. Oh, you have, Victor. Go ahead.
I have a question also. So in one of the slides you mentioned, Lindsay, that you plan to experimentally validate the motifs that you are finding. Yes, they're the fourth point. So how exactly would you do this? Because I think that's a really important question. Yeah.
That is a really important question, and I have to say, this is something we've wanted to do for a really long time, and it's a barrier because I don't have a wet lab, and I have to rely on experimental collaborators, and also with T-cell receptors, I think it's a bit more challenging. And Carnita mentioned earlier the question about HLA, and so it's a bit trickier to design the experiments. We have several ideas. One is first just using tumor lysate to see whether there's any reaction. So if we have like peripheral blood PBMCs from patient and we co-culture with tumor lysate, if we see expansion of any T cells and we sequence, can we score those T cell receptors to see whether they score positively with our model? We've also thought about doing something where we engineer T cells and have them expressed in like a Jurkat cell line. And then again, you have to have some complex experiments that I have friends who do these, so I'm probably not even explaining the experiments very well, but where you can then test specific T cell receptors. We have this data that I didn't present today, which is kind of more interesting to me because it's more recent, but I really wanted to focus on studies where we had used public data for this session. But we have this more recent work where we have been looking in the context of cervical cancer. And that one may prove to be a tad easier because there's the HPV human papilloma virus etiology, where we can also do some like tetramer based experiments where we can hypothesize peptides from the HPV genome. So this is like something I've badly, we really need to do it to really show whether our method is even finding T cells that are biologically meaningful. It's really, really important, and we really, really want to do it. And it's just something that's been harder to get off the ground due to, you know, my not having a wet lab. But yeah, thanks for asking about that, so I could talk about it for a minute.
Hey, thanks so much. Yeah, very nice talk.
Very nice presentation and very nice settings for the machine learning. It's impressive. I have a question. So you are looking at finding these very small motifs. How could you find them elsewhere in other unrelated data sets, just to see how frequent they are in the general population and whether there could be predictors, more general predictors?
Yeah, that's a really good question. So we haven't really done that, primarily because we assume that with such short motifs, they're kind of guaranteed to be present in the general population. And I don't know if I stopped sharing my screen or if it's still sharing, but I don't even.
Sharing.
Oh, I did. Okay. Well, part of our model involves also utilizing the receptor abundance. And so one thing that has proven to be an important aspect in our approach, where it may be that you find these motifs within the CDR3 sequences in control populations, but their abundance is much smaller than what you find in the case populations. And so having that abundance feature is something that is important to us. When we go without the abundance feature, our leave one out cross-validation accuracies drop down into the 80s rather than the 90s. And our test set validation, our test set accuracies drop down to in the 70s rather than in the 80s. So the abundance is an important part of the performance for us.
Okay, yes, that makes sense.
We could lengthen the motif. We haven't done that because our sample sizes have been so small. We have wanted to keep the number of parameters in our model also very small to reduce the risk of overfitting. But once we have our much larger data set, I think we can be more permissive at looking at longer motifs, or even looking at multiple motifs, having multiple detector functions that would detect multiple motifs. So again, maybe next year I can show that.
The conclusion is that we need more data.
We need more data. We need more money, and we need more data.
All right, so we are already running a bit late. So we are supposed to have a break now. So I think we can keep. There's a question maybe from the chat. What do you think is the impact of patient HLA haplotype on finding TCR clonotypes, and do you try to have a diverse population of haplotypes in the study? So I guess it was for you, Lindsay.
So I think to what Victor responded in his talk, that I think Adaptive has done some nice work showing that you can find these motifs cross HLA. And even I think you can predict, I forget the accuracy, but I think that they have shown that you can even take someone's repertoire and from that make decent predictions about what their HLA type is. Someone correct me if I'm wrong, but I think they have shown that. Beyond that, that's kind of also one reason why we focused on CDR3 and didn't pull in also CDRs 1 and 2, which I think show more interaction with MHC. And we have also not done HLA typing on our patients, although we do intend to going forward in this larger study, where we may be able to include HLA as a feature in the model. So we didn't intentionally diversify HLA in our populations, but HLA is unknown in our cohorts. We did have racially homogeneous populations. So that may, to some extent, restrict the how much variability there is in HLA. So for the ovarian cancer, we used a non-Hispanic white patients exclusively. For the cervical cancer study, we used Hispanic white patients exclusively. And again, in the larger study, it will be racially diverse and ethnically diverse with known HLA type.
All right, thank you, Lindsay. So
Thank you.
let's have a 12 minutes break so that we don't run too much late on time, and we'll join in 12 minutes back with Nina as the next speaker. Thank you.